Zinc is an important and essential element that is needed for the proper functioning of different aspects of life. On a broad surface level, zinc is associated with many different health issues such as anemia, schizophrenia, loss of appetite, neuropsychological changes, and other conditions. Additionally, zinc is involved in signaling, where free zinc ions are released through transporters by specific cells such as neurons. ZIP12 is one of these transporter proteins and is highly expressed within the brain, as well as in astrocytes, which are glial cells that have multiple functions, like supporting signaling. While we know and understand that zinc is co-released with glutamate, we don't know how zinc is cleared, and the relationship between ZIP12 and astrocytes may explain this. It is hypothesized that ZIP12 is involved in removing zinc from the synaptic cleft into astrocytes. Using high-resolution microscopy, it is possible to see how ZIP12 and astrocytes interact with each other through looking at the stained brain slices of the auditory cortex in mice, as well as to determine the localization of ZIP12. Through this imaging and staining, it is possible to determine where ZIP12 is in relation to astrocytes. The part of the images on this presentation that are in green represent the astrocytes, and the parts of the images that are in red represents the ZIP12. These images are taken using two different channels that pick up on different stains that are later merged. In the bottom of the left column, those two pictures show how these different channels work separately, and these two images are later merged together into the image at the bottom of the middle column that shows an overall broad picture of the location of the astrocyte and ZIP12s. The places where there is a direct overlap of these two colors on the images show a colocalization of ZIP12 and astrocytes, which shows an association between ZIP12 and astrocytes, as well as a step in proving the overall hypothesis. Preliminary results such as this colocalization has shown that an association between ZIP12 and astrocytes exists, and future work on this subject will focus on confirming the relationship between ZIP12 and astrocytes, as well as better understanding the role ZIP12 has in the brain and how this may impact how the brain functions.